ありがとうございます。Thank you for the introduction. 皆さん、こんにちは。オンサイト、オンライン。私はチュンです。8年前、日本来ました。今は理研のティームリーダーです。と、広島ユニバーシティの visiting professor。今から英語で。That's all I have. All right, so I'll start with some introduction of myself, and then I will talk about where I work, which is Weekend, and then I tell you what I do and who are we looking for at the end. So a little bit of a history of myself. Who am I? So that's pretty much summarized my 22 years journey as a researcher. So I know a lot of you now is considering whether you want to be a graduate student or not, so that you might take it as a example of how I went through my journey. So I graduated 20 years ago as an undergrad in the University of Hong Kong. I was working on viruses at the beginning, and then I moved to France to work on amoebas, which is a parasite, as a postdoc. And when I come here 2014 as a postdoc, and start working on human transcriptome, and 4 years earlier that I became a team leader here at Weekend. So I was originally as a molecular biologist, but later on became a computational biologist. So that's sort of sets an example that you could change your career path at any given time point. So it's not what you study, is what you're interested in. So where do we work? I'm from Weekend, as I said. There are 4 divisions in the Yokohama campus, Tsurumi, in the center of IMS, that we are in the division of genomic medicine. So the main theme of this division is we try to explain how disease happened from the DNA perspective or from the RNA perspective. Apparently, DNA refers to genetics. RNA is our major work, which is on looking into the transcriptome of different disease conditions. So, so what exactly do we do? Okay, so let's start with some basic questions. So when you look into the population, everybody is different, right? So some of the people, it's easier to get diseases, say, for example, heart attack in this case. Some of the people are less likely to get heart attack. And then we ask the question, and what exactly is a factor that is contributing to this kind of differences between individuals? And of course, that deals with the fundamental differences between individuals, which is DNA. So actually a long time ago, 20 years ago, that the very first study of looking into the DNA differences between cases and control, in this case is a heart attack, was actually performed at Weekend 20 years ago. So, and it turns out that you have a list of variants that is associated with diseases. And so there's good news and bad news. So the good news is there are certain variants associated with the diseases, but the bad news is a lot of these variants is not residing in the coding region of the genome. So and actually 98% of those of the regions is not coding in our genome. So it is very hard to understand what exactly these associated disease-associated variants are doing. So, and in fact, if you really look into the distribution of these disease-associated variants, and the majority of them, it is actually located, which is about 60%. You look at all these circles right here, these red boxes, which is about 60%, is located in somewhere we call the regulatory regions in the genome. So what exactly are these regulatory regions are doing? So we refer as promoters and enhancers. So these regulatory regions, and by definition, they're regulating the transcription of genes. So a lot of disease variants is actually located in these regions, and they were not actually changing the gene themselves, but they're actually changing how the transcription of the genes is regulated. And when this regulation happens, then you have the disease, or you're more likely to get the disease. So this is the rationale of everything. So So to look into the activity of these regulatory regions, well, there are many different methods, but in our group at Weekend, that we are more focusing on detecting the RNA, the transcriptional output from these locations. Here, that if you're interested to look into more details, you could look at the papers. But the idea is by measuring the 5' end of the RNA molecules, that we could precisely look into where does the RNA came from, and hence to pinpoint exactly where the transcription regulatory regions are. So, but unfortunately, all these transcription regulatory regions, they are active in a very specific cell type. So here I'm only listing a few. And so that's why looking into the activity in single cells is really important. So here I'm just giving you an example that on the left-hand side that you see that if you measure the activity within the bulk tissue, that everything is mixed together, that you might not be really finding the important signals from the cell type that is really related to the disease. So on the right-hand side that you're seeing is the breaking down of the tissues into single cells. So you could isolate the signal that you get from one particular disease population. So that comes to the question that that how do we get the signal of transcription in one single cell at a time. So here is a common technology that we use in our laboratories, which is encapsulating every single cell into a little droplet, and we make a single cell RNA library from that droplet, and then we could sequence what's in the cell in order to map this cis-regulatory element activities. So we've done so with 22 different tissues we collected from multiple individuals, and altogether we sequenced 460,000 cells and devised over 200,000 cis-regulatory elements in the human genome. And so these maps that you're looking into, every single dot on this map is one single cell that we sequenced. And how do they position on the map is based on the activity of cis-regulatory elements. So, and of course, based on various, clustering algorithms, that we could define something that we call cell classes and cell types. So this is a little bit overwhelming right here, but we could look into specific examples and highlighting why we are doing this. So our purpose is to look into whether there are cell type specific activity of these cis-regulatory elements is actually enriched in the disease-associated SNPs, which is the hypothesis that I put up earlier. So here I'm showing you one particular example. So on the left-hand side, that you're seeing only a very small clusters of cells. All these cells are blood vessel cells. We call them endothelial cells. The top plot is highlighting by the cell type, and the bottom plot is highlighting by the origin of the tissue. So when we look into particular diseases, say for example in this case is a disease called varicose vein, which is swelling of the veins on your leg. We asked the question that how much of the genetic association signal is associated with each of those cell type specific cis-regulatory elements. And we find out that on the center plot here, that the more red the color, meaning this cis-regulatory element is more enriched in the disease-associated SNPs. So we find out there is only a very small specific type of endothelial cells, which is actually muscle-specific capillaries and muscle-specific veins are enriched to the varicose veins. So this basically points out that these disease predisposition is very cell type specific. So let's switch topic a little bit. So that project that I was describing was pretty much, going towards the end. Here that we have another project which is ongoing. If you guys are interested to come into our group, that might be the project that you guys will be working on. So it is about ALS. The idea is that whether we could use an in vitro system to use the iPS cell lines derived from patients, and we program into motor neurons, and we look into the differences between the control group and the patient's group, and to look into on a single cell resolution. So to pick up these regulatory elements that may be overlapping with the ALS SNPs. So let's go to the end right here. So who are we looking for? So we are looking for someone who loves science. So if you love science, drop us an email. And if you're interested in computational biology, programming, statistics, sequencing, genomics, transcriptomics, or if you're interested in experimental biology, in particular neuroscience, cell culture, iPS differentiation, drop us an email. So this is the end of my presentation. Thank you very much.